last of our thanks. This is the last of our CCB skills seminars for the semester. Next semester, we're planning on doing a similar format on Zoom. We're still rounding up speakers as well. So if you're interested, just keep on our mailing list and keep a lookout for a registration RSVP Google form at the beginning of the next semester, just like we did this semester. Yeah, thank you for those of you who are here and have continued to participate. Today, we're hearing from Adam Gayoso, who is a graduate student in the Joseph Lab at UC Berkeley. And he does a lot of work on single cell analysis. He's kind of like the resident expert in the lab on various single cell stuff. He could have probably conversate like a literature review off the top of his head instead of today doing this actual tutorial. But he's a great person to work with, and I think he's going to have a lot of really great stuff to talk about with regards to SCVI, which is one of the toolkits that the Joseph Lab has put together as kind of like this general analysis platform for single cell data analysis, as well as for Scanpy, which is a Python-based library for doing basic single cell analysis. Yeah, so without further ado, Adam, why don't you take it away? And actually, I just want to say one more thing. We have a Google form that we're going to send out after this. If you could just let us know and give us any feedback on that as well, that'd be fantastic. But yeah, sorry. Now you can take it away.
Yeah, thanks, Matt. That was probably the best intro I've ever gotten. So anyway, thank you all for joining. I created like a little website here on our CCB skill sound that I posted in the chat. And on this website, you can navigate yourself to the Colab notebook, which is how I'll be running code today. There's also an HTML version that you can just, if you don't want to like deal with Colab right now, you can just click on the HTML version. And today I'm going to be talking about sort of just general purpose analysis of single cell RNA sequencing data. And I'm going to be highlighting these two packages, Scanpy and scvi-tools. And I think this is a general purpose tutorial. So maybe if you're not familiar with single cell data at all, you'll become a little bit familiar about like these objects, what the data looks like, and how people analyze it. If you're more familiar with an R-based pipeline, hopefully this presents the Python-based alternatives. And even if you're very experienced, maybe you'll learn a couple of things about the way I approach things. So I encourage everyone who has Colab open to start running through the first few cells. So especially this package installation cell, which will take like 2 or 3 minutes. And just to give you an idea of where we're going to go, there's this really great paper from the Tice lab called Current Best Practices in Single Cell RNA-seq. And I think this is a really good paper because it anchors you in a certain way about what are the sorts of things that people are doing to analyze single cell data. So I've been kind of working with single cell data since about 2016 on and off. And when I started, I kind of wish that this sort of resource existed because people did all these things to the data and I wanted to know why. And I think this paper does a really good job of motivating a lot of the things that are done. And this is the figure 1 from the paper. And today we'll be mostly focusing on this left side. So like going from a count matrix to doing some basic quality control, batch correction or integration, and then clustering and annotation. There's this whole other field of trajectory inference and understanding continuous relationships in the data. This is probably worthy of another tutorial, maybe next semester, Matt. We could focus more on this because there are hundreds of tools for finding these pseudo-time trajectories. So anyway, yeah, let's get started. So I think I should have run the first few cells. So I think Colab is a really great environment actually for doing quick single cell analyses. To install these packages, it actually takes like less than 2 minutes. And even with the fact that Colab will time out after a couple hours, it's still pretty quick. And one of the other reasons Colab is really great is that it gives you access to a GPU. By using the link that I sent, it should be the case that if you navigate to this runtime and change runtime type, that the GPU hardware accelerator is being used. And if it's not, perhaps you want to switch it to GPU. But and the reason is our scvi-tools models run a lot faster with a GPU. And Colab basically just gives you a free GPU to access, which is great. So now that the packages are installed, I'm going to download some models that are pre-trained that'll be relevant later. This is just to save some time in this tutorial. And finally, I'm going to import the packages that we're going to use today. So mostly it's these three packages. So ANData is the dataset object that I'll go in depth with. scvi is the package that we'll use to integrate datasets. And Scanpy is sort of this general purpose pipe library that would be similar to Sarot if you're familiar with R-based analysis. One of the things that you might want to do when you first import Scanpy is configure some of the global settings. So I use Scanpy a lot for visualizing things, and you can do this sc.set figure params, and I can set the fig size. And I can also tell Scanpy that I don't want like the frame around particular figures, and that'll be more clear later. But basically what this does is it changes like the matplotlib RC params globally for you. Scanpy also has a way to set the number of jobs. I think on Colab, there's only 2 cores anyway, so just setting it to 2 should make some of these steps a little bit faster. So the data that we'll focus on today is data that was actually measured using a technology called CiteSeq, and CiteSeq is single cell RNA sequencing plus an extension to measure proteins, and this would be a paired measurement. We're going to ignore the proteins today, so we can just think about this as a single cell RNA-seq data set. So let's go ahead and download that from scvi. We actually host this data set in the scvi package. Didn't run import. And essentially what this is doing is just downloading a file that we've hosted. It's most likely the case that you're going to have data that's not already formatted as ANData, and Scanpy actually has a number of ways to load this data. So if you just like look at the Scanpy API, one of the things I really like about it is that the documentation is really nice. And as you can see, they have basically a way to read from all the popular formats, like from 10x cell ranger output, from a loom file, which actually allows you to go back and forth from R and Python, just from a basic CSV. And every time you use one of these functions, it returns this object called an ANData. So we can think of an ANData as a fancy data set that allows you to store metadata along with at the cell level and the feature level, which would be the genes. This is kind of a representation of this object. It's kind of anchored by this X attribute, and this would be your cells by genes matrix. One of the important things when you're doing analysis in Python is that it's always cells by genes. If you're coming from R, you'll often see that it's genes by cells. So in Python, it's always cells by genes. And we have these 2 pandas data frames kind of attached to this X matrix. So we have it's called .obs, and this would be a cell level metadata pandas data frame. And similarly, we have .var, which would be a pandas data frame that you can add metadata for each of the genes. And finally, there's this unstructured, which I won't be using today, but you can basically throw anything in there. So it's a dictionary, and you can throw just random NumPy arrays or other metadata that maybe isn't at the feature or cell level. So now in the notebook, I'm just going to show you some of the things that you can do with this ANData object. And even if you're not someone who does single cell analysis a lot, I actually find this ANData to be have a lot to be really good just generally, because it's easy to store a lot of different data in one file, and then you don't have to fish around for 10 different files. So I made a copy of the ANData that I downloaded from scvi, and I just called it B data just to make it clear. And just doing some exploration, let's look at the observation level metadata. We see that the index of this pandas data frame is the barcode for each cell. And we have some metadata that came along with this data set that was processed while the data set was being analyzed for the publication, such as the cell type for every cell, which tissue it came from, which batch it came from. And batch here means, in this case, which 10x chromium lane the cells were processed on. They were also processed on 2 separate days, but that's not really that important of a detail for right now. Similarly, we have the .var data frame, and this right now is just empty, but the index is the name of every single gene in the data set. So I can use this thing called bdata.var names, and this will just return for me all the gene names. And this is important because when I start doing plotting, it'll use the names stored in .var names to plot the expression of genes, for example. And data is really flexible. This X attribute, which actually contains the count matrix, as we can see, it's 30,000 cells by 13,000 genes right now. This can be a sparse matrix. It can be a NumPy dense array, and so on. We can add new cell level metadata by using this, I guess, just this bracket operator. This is basically a pandas style way of adding data into a data frame. And I'm just going to add some dummy data where it's just the word test extended over all the cells. So .nobs would be the number of cells in the ANData object. So now that I've done that, I can see that I have this new column, new cell level data, and it just says test. And having this organized is very important when you start doing plotting with Scanpy, and we'll see that later. So now that we have some familiarity with the ANData object, we might want to do quality control in the data set. This data set in particular was already filtered, but I'll show you some of the things you might consider when you're doing basic quality control. One of the big important things that you might do first is compute the percentage of the observed counts in a particular cell that correspond to mitochondrial RNA. And that can be done by using this method called calculate QC metrics. And this is a Scanpy function where I tell it which genes are mitochondrial. And I did that using, this is a sort of a pandas way of doing that. I'm asking all the gene names that start with MT. And basically this will be, this should just be a Boolean like mask over the genes. So it will return true if it's a mitochondrial gene. So now that I've run this function, I can see what Scanpy did. It added new columns to the observate.obs data frame, like percent counts MT, total counts MT. And an easy way to visualize this is this function called Scanpy plot violin. And what I do is I give it the ANData as well as which columns in this observation data frame that I'd like to plot. And it's going to make these violin plots for each of these columns. And when you're doing quality control, you're kind of looking for outliers in either direction. So it's possible that I might want to filter some of these cells that have really high total counts, like this one has more than 60,000. It's possible this is some sort of doublet or other technical artifact. I'm not going to filter this particular data set, but usually, like for example, percent counts mitochondrial, you'll see like another mode even higher than 15, a small mode. But you would want to potentially remove those cells. And it's not always so simple which cells you want to remove. You might find that you do some conservative filtering and then you run your analysis, and then you find that there's weird clusters that might not correspond to anything biological that you know that might actually be technical in nature. And then you might revisit these thresholds down the line. By the way, if anyone has questions, feel free to throw anything in the chat or just interrupt me, honestly. So the great thing about Scanpy is there's little mental overhead. I don't need to think about plotting things so much because I can just run these functions. So if I just want to plot the total counts per cell versus how many genes were detected, this is the name they use from their quality control function. I can just run this Scanpy plot scatter, and I'm just telling it x-axis, y-axis, which information to plot from the data frame. And this makes analysis just a lot quicker when you don't have to start writing custom plotting code. If I did want to actually subset the and data object, it's pretty simple. I would just go and data equals and data, and I would just get the cells that have fewer than 15, than the percent mitochondrial is less than 15. And I can do that simply like that. You can subset the and data much like a pandas data frame. So like to that end, I could just do and data. I can make, what if I just want to look at CD4? This would return an and data object that has only one dimension for the one gene that I selected, but it has actually all the metadata associated with it. It's just that .x would be one dimension. So yeah, this one dimension. Yep.
May I ask a question about the scatter plot?
Sure.
Do you know what you would want to see? Like, is this what's indicative of a good scatter plot to see here?
I mean,
like, what's the curve supposed to look like?
Oh, I mean, these two things should definitely be correlated. Whether or not these cells at the top right corner are artifacts or not is kind of up to the analyst. I kind of recommend just being conservative at first. And it's kind of typically the case that technical artifacts, you'll find that they cluster together later on. And then you might want to remove that later. But like for example, you might not want to sort of filter things on the low end because that just might correspond to some cell type.
Oh, I see. Okay.
It's hard to know a priori what's technical and what's not.
So would you say it's better in general to filter out really high UMI counts or something like that versus something with really low counts?
Usually yes, but I guess it also depends on like the nature of the system that you're studying. Like if you're studying some activation or something, then maybe the counts, the high, you know, those could just be like not necessarily technical. So it's kind of hard either way, but usually if I hadn't pre-processed this data set, it's usually a lot more clear what to cut off. But in this particular data set, which we analyze in our paper, like we actually went back and forth on this a couple of times. And I think that's pretty natural.
Good to know. Thank you.
Although when you read a paper, it's like, you know, we filtered X and Y at this cutoff, and then like they don't really explain why. And that's just kind of how the field is. So the most basic normalization is certainly not a solved problem in single-cell RNA-seq, but just about every paper either uses this technique called log normalize. Some people use a method called scran, which is only in R. And then other people use this new one called SC transform, which is in Sarot. In Scanpy, the basic normalization is this log normalize. So the first thing that I'll do is I'll store the counts, which is currently in adata.x, in a layer. So a layer is, you can think of a layer on top of x. And we want to store the counts because some methods do require the counts, but a lot of them actually require this log normalized data. So I'll store the counts in a layer. I'll normalize the total. So this scales every cell to have the same exact sum. So everything will sum to 10,000. Then I'll log transform +1 because there's a lot of zeros. And then I'll store the and data in this attribute called raw. This is probably one of the most confusing things about a Scanpy workflow. It basically freezes a copy of an and data in this attribute called raw, and it's used a lot in the Scanpy plotting and other functions, and that'll be more clear later. So I'll run this. And now I have log normalized data in X as well as raw. So it's actually in 2 places. The next thing I'll do is highly variable gene selection. So this is the idea of gene selection is that I want to learn a distance metric between cells that kind of captures the biology. And if I compute distances using the matrix right now, I have 13,000 dimensions for every cell. So we have a problem with the curse of dimensionality. There's also just a number of genes in there that aren't really biologically informative. So this is the idea behind highly variable gene selection is I'm going to subset my data set to only have about, I think people typically take 1 to 5,000 genes I typically see. And the idea is that I want to pick genes that have a higher than expected variance. So Scanpy has this method called highly variable genes, and it's actually using the method of Sarot v3. So to give you an idea of what that does, Scanpy has this plotting function where I can plot the output of this. So what's happening is if I plot the mean expression of genes on the x-axis versus the variance, we see this relationship that the variance scales with the mean. I want to pick genes that have a high variance kind of controlling for the mean expression. If I pick genes that just have a high variance without looking at the mean expression, then I'm actually just going to be biased towards genes that have high mean expression. So that's kind of what the Sarot v3 method is doing. I like it in particular because it operates directly on the count data. I think it's also probably the most common method used right now. But so how it works is that we have here the variance of genes on the Y and the mean expression. And what it does is it takes a variance stabilizing transformation of the data. So it kind of transforms the data to control for the mean expression. And then it's a little bit more complicated than that, but what you get is what you see here, where the variance doesn't seem to depend so much on the mean expression of the gene. This would be after the transformation. And then basically it's taking genes, the most highly variable genes, you can think of just looking at the y-axis and going from top to bottom. In the Sarot workflow, they usually use 2000. I think higher is usually better, but there's probably like strong diminishing marginal returns after a while in terms of how informative each gene is. So now that we've done that, I'm just going to subset the and data object to the highly variable genes. And that can be done by passing, this is a mask on which genes are highly variable or not. And I can just subset the and data object like that. I'm putting .copy here so it returns a copy instead of a view. This is a subtlety of and data. It's kind of necessary for running scvi. So now that we look at our and data object, we have 30,000 cells by 4,000 genes. But kind of going back to that .raw thing, .raw still contains all 13,000 genes of the log normalized data. So I can run adata.raw.toA data and it returns an and data object that I can always just recover. So raw is kind of like frozen and protected in a sense.
Hey, Adam, can I ask a quick question?
Yep.
What's the purpose of the copy function? I see that you threw that out there. I mean, this is very like minor, but like, why even have that? Why not just subset without the copy? Is there something that it does?
So and data has this thing called view, and basically if I didn't do .copy, it will return a view on the same object, just subsetted at those genes. And it's just a subtlety of when you run a method like scvi, it needs a copy. I'm pretty sure actually if I just kept going on and didn't have this copy, at some point it would copy it in memory, just behind the scenes. So I think in the scale or PCA step, it would actually just do it. I just like to be explicit.
Hey, Adam, I think it's, correct me if I'm wrong, it might be pandas actually, that it's not the A data itself, but it's underlying panda objects. And if you're working with a view, you're modifying the underlying panda objects rather than what you think you're underlying, which is a copy.
Yeah, I mean, definitely in Python, I always try to copy when I'm sure I don't want to alter what was happening before. Like I've had issues with and data where I thought, like if you initialize an and data object with data, and then you edit, and then you have like a variable A and then the and data, and you edit one of them, you're actually editing both of them because it's pointing to the same memory. So it's just good to be explicit about about these things, I think.
Exactly. Yeah.
Yeah.
Yeah. You can also see in pandas.
So let's dive into the standard workflow. So this would be the most basic thing you do. So the first thing is dimensionality reduction. So even though we've reduced to 4,000 genes, it's a lot simpler to work with, let's say, 30 latent dimensions, I'll call them. And the idea is that even though there are 4,000 genes, the genes are highly co-regulated, so I can probably use much fewer dimensions to explain most of the variation in the data, and that's what PCA is doing. I won't go into depth with PCA, but one of the steps that most people do before running PCA is the scale. So what this does is standard scales each gene. It kind of weights them equally when you're doing a PCA. Otherwise, PCA might be biased towards genes that are highly expressed. So I'll run this. And the way data works, it's very similar to Sarot in this way, is that when you run PCA, what happens is it actually stores the output in this thing called .obsm. And .obsm is a place to store a bunch of matrices that have the same number of cells. So andata.obsm x PCA, it has the same number of cells as .x, which is 30,474, but it has 50 dimensions. And I can store any number of matrices with a key name in .obsm. So Scanpy by default will use this XPCA matrix to compute nearest neighbors, a neighbors graph between cells, for a cell by cell neighbors graph. So Scanpy actually uses this approximate nearest neighbors method that's implemented in Umap. So they've actually sort of decomposed Umap behind the scenes. It's not that important of a detail, but it's pretty fast. So right now what's happening is I'm computing a neighbor graph where the distance between two cells is defined as their sort of Euclidean distance based on these principal component scores. And that's done with scanpy.preprocessing.neighbors. And I can specify like how many neighbors I want. I can specify different distance metrics. I think some people prefer the cosine distance. I think I saw somewhere that there was a paper that evaluated these, but I can't remember exactly right now. So now that I have this Umap representation of the data, Umap is just a way to take the 30 dimensions and kind of compress it down to 2 even, non-linearly. So andata stores the Umap in another one of these obsm in the obsm field. So now this is only 2 dimensions, and I can just plot it, and I can pick colors from my metadata and just color the Umap by that. So here I'm coloring by batch. I kind of described this earlier, but this data set contains 4 of what we call batches. So on day 1, the blue and the green cells were prepared on separate 10x lanes, and on day 2, similarly, they were prepared on different 10x lanes. We can see that there's much stronger of a batch effect between days than between lanes on the same day. So now that I've gotten here, I realize I have pretty strong batch effects in my data, but I actually want to find sort of biological variation that's consistent across these batches, and this is what data integration is called. Scanpy has a number of methods that actually do this for you. One of them is called BVKNN, so I'll just run that right now. And you can go through the Scanpy docs and sort of, they have this external API. So if you're someone who likes to develop methods, you can submit your method to Scanpy, and they host a number of these. So Harmony is another really good integration method that is popular in R, but there's a Python implementation here. Similarly with MNN, which is probably like the first modern single cell integration method. And there's just a number of methods here that are worth trying out. I already see that time's going by quickly. So I'll start talking, while that goes, I'll start talking about SCVI. SCVI is this method that was developed in the Joseph lab and has since sort of evolved into this package called scvi-tools. And we can think of ourselves as taking like a tangent off of the Scanpy workflow and using scvi to accomplish this task of data set integration. So I ran bbknn, and like we can still see that it didn't really mix the batches very well. That said, from what I understand, bbknn scored really well in this benchmarking paper. So I think it's a good method, but sometimes maybe it's good to try a couple methods out on your data set. So moving into scvi, scvi kind of encapsulates many parts of the pipeline. I think it really excels in this dimensionality reduction and integration setting where you're integrating data sets from different experiments, different labs, different species. It also has this differential expression feature, which I'll describe how to use here. So we can kind of think of scvi as a nonlinear alternative to PCA. There's a lot more technical details. I won't go into depth, but basically we have our data set that's cells. Here it's actually genes by cells, and then we encode it using a neural network into a matrix of factors by cells. And this would be the low dimensional space that's also integrated. So we can, instead of using PCA, we can use scvi, and it's kind of a drop-in replacement, but it will also integrate the data sets for you. On the other side, we take this low dimensional embedding and we try to blow it back up and reconstruct the data. And scvi is a Bayesian model, so we can sort of leverage the uncertainty over all these things and actually try to understand if a gene has a differential expression between two sets of cells. And if you're not familiar with differential expression, I'll define what that means a little bit later, like in the next 2 minutes. One of the important things to run scvi is this function setup and data. So the input to scvi is an and data, but we have a variety of models in the package, and you need to tell scvi which parts of the and data you would like to use. So for example, my and data had log normalized data in it, but I stored the counts in a layer. So I'm going to specify that the counts are in a layer called counts. And the batch information for doing integration is in this metadata key called batch, which is what I had just visualized here. This is the batch key that was in the OBS data frame. Once I've done that, I can get a visual confirmation of what scvi did. I can run this function view and data setup, and it kind of tells me it's going to take the data X from this particular location in the and data, and that just gives me, makes me a little bit less stressed out about what's going on. So, because there's a lot of things going on here. I can also see I'm doing integration with this key batch and data.os batch. It gives me the categories, and scvi has to actually encode these into integers to do batch correction. So it tells me what integer it's going to encode that. Now I would ordinarily train the model, but I pre-trained it just to make things a little bit faster. I think this data set would take on the order of 10 to 15 minutes on Colab. So I just loaded, I had this directory that I downloaded early on, spleen lymph scvi. I'm just making a model. This is the interface at this point is very similar to scikit-learn if you've used that. So I've loaded my model, and I can actually, the model has a representation that you can print. So the scvi model is telling me that it's been trained. And now what I want to do is I want to get the latent representation out of the model. That's this batch corrected integrated latent space. And I'm just going to store it back into the and data object, similarly to how PCA was stored. I'm also going to store this sort of denoised expression out of scvi. There's a lot of literature now, or a couple of papers that describe whether people should be using these. This would also be called like imputed data. I can refer people to that literature. It's an interesting conversation. We typically use it at this point now just for visualizing data, and I'll show you what that looks like. So now I want to compute a Umap using the scvi latent space. So I have to tell Scanpy that I'm going to compute a neighbor graph using the and data, but using the representation X scvi, which is exactly how I stored it. I'm going to use a different distance metric as well, the correlation distance. So I'm going to run that. I'm also going to, this is how you cluster with Scanpy. So it's called sc.tools.Leiden clustering. And it uses the neighbor graph that gets stored after this method. So once that's done, I'm going to plot the Umap with, well, you can already see it here, but this is going to reappear. We can see that scvi has corrected the batches. These cell types are cell types that came from an independent analysis from a paper that I was working on. We didn't actually use scvi, we used a similar thing. And we can see that it seems to have done something reasonable. And just to show you what the clusters look like, they kind of resemble the cell types. One of the cool things you can do in Scanpy is, once this reappears, is actually recluster. So like maybe I want to get more resolution on these myeloid subsets. So I can say Leiden clustering, but I'm going to restrict to cluster 14. And then I can just plot that again. And there's more, now there are more clusters here. So that's a way to actually dive into one cluster and sort of recluster it. I would be kind of careful with this, because you can arbitrarily keep reclustering things, and I'm sure you'll find something. Whether that means anything is kind of up to you. And often the way people find whether these things mean anything is differential expression. I'm not going to go into this math, but I'm going to give you like a sort of pictorial representation of what differential expression is. So imagine we have these two clusters in our Umap, and what scvi does is it gives you a distribution over this sort of denoised expression. So for example, in group 1, let's say the expression of gene CD4 follows this distribution, and CD4 in group 0 follows this distribution. What we can actually compute with scvi is like a posterior probability that the log fold change of expression between these two groups falls outside some region. So I want to know what's the probability that the log fold change of expression between group 1 and group 0 for CD4 falls outside of the region minus delta to delta. So we compute this using Monte Carlo samples. I'm happy to talk to anyone who's interested more in this method. This actually takes 2 minutes, so we can talk about it more. I'm going to just run a couple of these cells in the meantime. So because scvi is a Bayesian method, we can actually infer like this posterior distribution over the log fold change of expression of one gene over 2 groups of cells. You can use this to understand, to rank genes in a 1 versus all setting. So I might want to take cluster 0 and see what sort of identifies it as relative to all the other clusters in the data. Similarly with cluster 1, but I might also have cluster 1 where I have 2 conditions or 2 tissues, and I want to understand, is there differential expression within this cluster between these 2 conditions, which might just be a tissue. And you're fully capable of running all these things through scvi. There's this function called differential expression, and I'm telling it to group by the cell types. So this is doing a 1 versus all differential expression between cell types, taking one cell type and comparing to all the others and iterating over all of those. You can also specify like group 1 and group 2. You can specify the indices of these groups and so on. So in this, it's going to give me a data frame where it's kind of concatenated to be really long, and it tells you which comparison was done and all this other information that unfortunately I won't have time to go into. I can take the comparison between groups. I can take one group and compare it to all others. That's how you would do it here. I'm just going to let this run in the background and go back and describe what the data frames mean. Scanpy also has a differential expression method that's similar to what Sarot has. They call it ranked gene groups. Similarly, I would tell it group by cell types, and it would do a 1 versus all differential expression. And I can access the data using scanpy.get.ranked gene groups data frame, and I tell it which group I want. So this would be Tregs versus all others. And the score would be here, it's a t-test statistic. The log fold change would be the log fold change of expression. And this would be like a FDR controlled p-value. Once this is done, I'll describe what the columns in the matrix mean. Okay, so SCVI outputs this data frame and it tells you the probability that the gene is differentially expressed, the probability that it's not differentially expressed, and the probability that it's differentially expressed is kind of the area under the curve outside of the black lines region. So like to the right of here and to the left of here. The base factor in this particular case is the log odds of these two things. It gives you other information, like this would be the denoised expression of group 1, the denoised expression of group 2. Gives you summary statistics of the log fold change distribution, so the mean, median, standard deviation, min, max. This would be the mean of the raw counts. This would be how many cells were non-zero in group 1 and group 2, the percentage of the cells. This would be like library size normalized expression. And it tells you which, so this was activated CD4 versus all the rest. And this is a really long data frame and it has all the comparisons in there. So you can sort that just using Pandas API. So kind of moving on, I just want to show you how Scanpy, the thing I love about it is really visualization. And it really makes these really nice dot plots and heat maps. Just in the interest of time, I probably won't run these cells because the output's already here. But I took the SCVI differential expression results and I kind of sorted for genes that have high log fold change and genes that are significant. One of the important things, I guess, of this sort of analysis is that the p-values are kind of almost meaningless because you're double dipping with the data. And I can share some references if people are interested in that. I think there's some work being done in the statistics community to kind of correct for this. But the ranks should be fine. So I kind of do some filtering here, and then I'm going to visualize the top genes for every cell type using a dot plot. And what this is showing you is the circle is like what fraction of the cells in this group actually have non-zero expression. So they have expression. And the color is sort of how highly expressed it is. This gives you like an understanding of what are the main markers for every cell type. So IFIT3 T cells, I imagine these have high IFIT3. This is using the SCVI differentially expressed genes. You can also use this matrix plot, which is similar to a dot plot. It just doesn't have this idea that the dot should represent how expressed a gene is in a particular group. Just because there are zeros doesn't mean it's actually not expressed. We have a sampling and noise in single cell RNA-seq data. So matrix plot is just another way to visualize that. These are actually the same genes. And the functions are pretty simple to run. You tell it which column in the metadata you want to group by, which layer of expression. So here I'm actually using the SCVI denoised expression. That's what's being displayed here. I could use the log normalized expression. They have if I just had commented this out. I ran the Scanpy t-test behind the scenes here while we were looking at the other plots, and I can actually, they have a function where I can plot ranked gene groups. Like it's plotting the output of that and taking the top 2 genes per group. And you can do these really crazy things like make these stacked violin plots. I'm not sure this is helpful for anyone, but you can do it. This particular example, I think if you had a more controlled example, it would be more helpful to look at, but this doesn't do anything for me really, but it's cool. One of the other things you might want to do is visualize the expression of genes on the Umap. So on the top row, I'm saying plot Umap. I give the color. These genes are in the andata.var names. And by default, it's going to use the expression in the .raw by default. I can tell it to use a layer. So here I would be visualizing the SCVI denoised expression. And for these 2 genes on the right, it kind of looks the same. Maybe this gene on the left, there's potentially more contrast here. I think in general, using these imputed values for visualization is probably the best thing to do with them. I'd be careful using these denoised or imputed values for other tasks.
Yeah, Adam, we have 2 questions. Or I have a question. There's a question in the chat. I'll ask the chat question first.
Sure.
Would you trust the heat map or dot plot more as one counts 0 and the other one doesn't?
I kind of like the heat map more because a dot might be small just because it's a lowly expressed gene and you have a lot of sampling noise, so you observe a lot of zeros in a particular group. And I think that the matrix plot kind of, or a heat map, they actually have another function called heat map that's slightly different than this, is maybe a little bit more faithful to that property of the data.
And the other question is, why do you think that the scvi expressions are like an order of magnitude or two less than the raw counts?
Right, so that's because the default output of the scvi expression is scaled to sum to 1, and these were scaled to sum to 10,000. So you could scale, there's an option in the function where you can scale these to 10,000.
Gotcha.
Yeah. Okay, this is actually as far as I thought I was going to get, but Scanpy has all the, like, their documentation is very, very good. And I think once you get a feel for the AND data and sort of the workflow, it's easy to start plugging your AND data into a lot of these functions. This particular data set, we measured cells from 2 tissues, the lymph node and the spleen. It's possible that you might want to understand cell types where there's a higher density of cells from the spleen relative to the lymph node. They have this fun function called like plot embedding density. And it kind of gives you this density where it's showing you that maybe in this cell type, there's a lot more cells that arise from the spleen versus the lymph node. And you can kind of look into different cell types with this. And it's just a cool way of visualizing the data. As opposed to this would be the regular scatter plot. Like, it's hard to tell density differences exactly from a Umap representation. So, I mean, there's 3 minutes left. I had this whole part about scvi. I can just describe what scvi is, and then maybe we can wrap it up. So now imagine you've gone through this whole workflow and you've annotated your data. And now you receive new samples from your collaborator, and you'd like to annotate those samples. So scvi is really good for that. What I would do is I would take my data set that's annotated, and I would sort of join it with the unannotated data set. I tell scvi which cells are annotated and which cells aren't annotated, and I can transfer the labels in an automated way. The method is a bit more complicated than scvi, and their paper should be out soon, so you can read more about it. There's a preprint out right now. And just to give you an idea, is I took half the data, I treated it as unannotated, and then I asked scvi to automatically annotate the unannotated cells. And this is the output. So these were the original cell types. And this is the output by scvi. The colors look a little different because one of the, scvi has a little bit of a problem. There's one cell type that only had 9 cells. So it's hard for scvi to predict that cell type. That would be the plasma B cells, but otherwise everything else mostly checks out. And like, here's like a confusion matrix of sorts to look at that. And to run it, it's quite similar to running scvi. And that's one of the things about scvi-tools is it's pretty consistent across models. Like, if you get site-seq data that has protein data with it, it's a very similar API, you just run a different model. So in that sense, it's like scikit-learn. And on that note, I guess I'll just end here and take questions. And I can stay on for a couple minutes after too.
Yeah, I have a question, but then also I just want to note for those of you who haven't yet, please can you fill out the form that I linked in the chat, because that helps us secure funding for future seminars and also lets us know what you are interested in learning about and how we can get better as well. So that's in the chat. But Adam, I was curious for Scanpy, how does it handle novel cell types? Let's say you get a new data set that is mostly overlapping, but has a new cell type that for some reason you didn't observe in your first data set.
Yeah, I mean, I think Roman had worked on that a little bit. I know Gallon has run, Gallon, I don't know if he's still here, but I kind of imagine that those cells would have just high entropy, but I don't know that, I'm not 100% sure if scvi can actually like say that it's unlabeled or say that it's like unknown or something like that. But I think if you just like inspect the prediction, maybe it just has a high entropy across classes, then perhaps you can say there's something different about it. But if Gowan's on the line, maybe he knows, but
yeah, that's a helpful answer. We have some other questions in the chat as well. Someone's asking if you could send it to them separately, the imputation choice paper, the paper you talked about, or I think you referenced a couple of papers that talk about whether or not to use imputed counts. I think people might be interested in those papers.
yeah, I can. I'll add those references to this this website. So if people are, I'll add more references here related to the imputation. Yes.
Yeah, and then someone has another questions about how to load a large Sarot object into Python.
Right. So for that, we have a tutorial, scvi in R, and there's a couple of ways. So there's this package called Zell converter, which is a bioconductor package. That's one way to do it. You could save it as a loom object from Sarot. That's the second way. You could also use reticulate. So what you would do is use the reticulate R package and sort of build the ANData object from R using the reticulate interface. And it's worked in the past for me. It sounds like if your object is 70 gigabytes, I'm not sure how well that would work. It seems like a loom or this is Zell converter might work for you. Zell converter. It's from the TICE lab, and it's a bioconductor package. And then I see like use loom or H5AD. Oh, sorry, I guess that's a response to that. Yeah. But converting back and forth, it's still not that fun, but it's gotten a lot better, I would say. So in our scvi and R tutorial, we do show how you would build an ANData on the fly, and you can pass a sparse matrix into the ANData constructor, like an R sparse matrix. And it has worked for me. Unless your H5 is the wrong version. As I think Matt experienced once.
Yeah, it's a very frustrating place to be.
Yeah.
Cool. Any other questions? Feel free to put in the chat or unmute yourself and ask. All right. Well, we'll upload the video to YouTube and post that on our website, and all these resources are going to be on the website as well. Thank you all for coming and participating. I definitely found this really useful, and I use Scanpy and scvi every day. So nice job, Adam. Yeah, and like I said, we're going to have a skills seminar for next semester as well. We're just continuing to finalize some speakers, and we'll send out an RSVP form when we have everything nailed down. So hope you can join us then. And once again, there's a form in the chat. If you can fill that out and let us know how we did.
Yeah, and please let us know if you have topics you want us to address. That would be great.
Yeah, exactly. Yeah, most of the topics that we cover are suggestions from people. So thank you.
Thanks, Matt, and thanks,
Yeah,
Adam.
thanks everyone. Thanks for joining.
Yeah, thanks. See you all next semester.